In Oracle, we have live classes, recorded classes. Recorded classes have added benefits of watching at your own leisure when you have time, and you can pause the videos and watch it. And then we also provide you with notes based on the latest syllabus and MCQs. Apart from the academic support, there's also personalized counseling sessions to give you non-academic support to make your medical school journey more enjoyable. We have three different kind of subscription plans: basic, standard, and premium, depending on your needs. You can avail them for 15,000, 30,000, and 50,000. Although we have offers going on already, but you can have an additional discount of 20% if you use my coupon code mentioned on the screen. So now let's begin with the class. Glycogen metabolism. So what is glycogen? I'm sure you have heard of this term before. It is described as the storage form of carbohydrates. Now, what do you understand by this term, that storage form of carbohydrate? So your carbohydrates in the body, they are not stored as glucose, but rather they are transformed into a different form, which is known as glycogen. So your carbohydrates are stored as glycogen. Now, what is glycogen? Glycogen is a branched chain homopolysaccharide made exclusively from alpha D glucose. This is alpha. Okay, now what do you understand by this? Firstly, it is a branch structure. Secondly, homopolysaccharide. Now, this term has three words. Homo means same, poly means many, and saccharide is your sugar. So when many same sugar units are joined together, they form a polymer which is known as glycogen. And in this case, this repeating unit is alpha D glucose. Now the straight chains of a glycogen, they have alpha everywhere. Where I had written alpha, somehow the PDF has changed it into A. I don't know why. So just assume that it's alpha. Straight chain has alpha 1,4 linkages and branch point have alpha 1,6 linkages. Okay. Now, although this alpha ND and alpha 1,4 linkage and alpha 1,6 linkage, nobody's ever going to ask you about these structures and all that. But for your understanding, just so that you're not stuck, what is all these terms? I'll briefly explain you this. What is alpha D glucose? Okay. So now your glucose, as you all know, that it is a 6 carbon compound. 1, 2, 3, 4, 5, and here comes 6. Okay, this has your aldehyde group, and this is your, and then you have all the hydroxyls placed here and everything. And then D and L, basically D and L, these are mirror images. These two forms are mirror images of each other. And mirror images are known as enantiomers. In isomerism in chemistry, you must have studied that the mirror images are known as enantiomers, and these two images, mirror images of the same compound, they are known as D and L. So this is the D form of glucose. This is D glucose, and in humans, majorly we have D sugars. Okay, D sugars in humans. So this D glucose, the exact mirror image of this will be the L glucose. Okay, so the mirror image, when you draw, these hydroxyl will come to this side and this one will come here. So this will be an exact mirror image. So this is what is meant by D-glucose. Okay, alpha D-glucose. Now what is alpha? Where does this alpha comes from? 99% of glucose in solution, 99% of glucose in solution form, it exists as a ring. Okay, I just showed you this structure here, this linear structure. But basically, in your body, it is predominantly water. So glucose in your body will be found in a form of a ring. This will cyclize to form a ring. I'll tell you how. This structure here, you see this? Something like this will happen. This here. Your carbonyl carbon, this carbon 1, which has the functional group, this will be attacked by the hydroxyl here on C5. C4 can also be involved. Actually, it can be any of the hydroxyl groups, but basically, majorly, it is C5. Okay, so this will form a ring. So this oxygen of the aldehyde is shared between C1 and C5. Okay, so this will form a structure, something which looks like this. Okay, now if your hydroxyl at C1, this is the carbon 1. If your hydroxyl at C1 is in trans, that is below the plane, this is known as alpha D glucose. So now you get it, what this alpha D glucose is. Although it is not required for you, nobody's asking the structures, but for the sake of understanding, I've explained all this to you. Alpha D glucose. What is alpha D glucose? Okay. So this is your alpha D glucose. Now you get it? So repeating units of alpha D glucose and which are branched, they form glycogen. So your glycogen molecule looks something like this. It is highly branched like this. So, so this is what your glycogen looks like. And why is it important? Because the enzymes in the body, they are able to recognize, they are able to differentiate between alpha D glucose and beta D glucose. Okay. For example, your glycogen is formed from alpha D glucose, but your cellulose is formed from beta D glucose. So here is the importance of knowing these forms, knowing the isomers. Okay, moving back. So glycogen is the storage form of carbohydrate. Glycogen is a branch-chain homopolar saccharide made exclusively from alpha D glucose. And the straight chain has alpha 1,4 linkages and branch point has alpha 1,6 linkages. Again, now it is very simple. You can understand what is alpha 1,4 linkages. When the two molecules that are making the linkages are in alpha conformation, it is alpha linkages. And when they are in beta conformation, it is beta linkages. So here it is alpha. That means the sugar molecules are in alpha conformation. And we already know because glycogen is formed only exclusively from alpha D glucose. So obviously the linkages will be alpha linkages only. Now alpha 1, so the carbon 1 of 1 and the alpha 4, that is the carbon 1 of one sugar of one glucose will form a linkage with the carbon 4 of the other one. Okay, and but at branch point. So when here these linkages will be alpha 1 4, but here at branch point, here all these branch points, these will have alpha 1,6 linkages. Okay. Now, what are the major sites? Where is glycogen stored? So it is stored in liver and in muscle. Now, what is the role of liver glycogen? Liver glycogen, it aims to provide glucose during fasting. Whereas your muscle glycogen, it provides ATP for muscle contraction, especially during exercise. These 2 points you will get to know in detail. I'll just explain you when we do lysis of glycogen. Now, liver has the highest percentage by tissue weight of glycogen, but the total quantity of muscle glycogen is more than liver glycogen because of the larger muscle content. Obviously, so it's saying that the highest percentage by tissue weight of glycogen is more in the liver, but since the muscle mass is more, the total content of glycogen is more in the muscle. Now, all of the glycogen metabolism enzymes are cytoplasmic. So that was a brief introduction. Now moving on to glycogen synthesis. So now we begin, which is also known as glycogenesis. Now, why is glucose stored as glycogen and not as glucose itself? So, you must be wondering that why to take all the pain of creating glycogen, synthesizing glycogen, why cannot we simply store glucose as glucose? So, it is because, as I mentioned, that your glycogen is a highly branched polymer. So, obviously, branching makes it really compact. And the other thing is that your water, it is so your glucose is water soluble, so it will absorb a lot of water and the cell will swell up. So these are the two reasons why you need to store glucose as glycogen. Now, glycogen synthesis takes in the well-fed state under the effect of the hormone insulin. Regulation of glycogen, we'll talk about it later. But for now, you should know that when is glycogen? Obviously, when there's excess glucose coming into the body, more than you need to synthesize and store it for later use as glycogen. Now, steps in glycogen synthesis. The first step is synthesis of UDP glucose. The next step, it is followed by glycogen synthase reactions. This is the rate limiting enzyme. And then comes the branching, which is the last step of glycogen synthesis. Now, the alpha D glucose attached to uridine diphosphate UDP is the source of all the glycosyl residue that are added to the growing glycogen molecule. Okay, I'll explain you this, what it says. Now, what is UDP glucose? What do you understand by UDP? First of all, tell me that. UDP glucose. Now, first, actually, let's discuss about UDP first. Okay, UDP is uridine diphosphate. Now, what is this uridine diphosphate? It is a nucleotide. What is a nucleotide? You'll talk about this in your genetics classes. But right now, you know what is a nucleotide. It has a pentose sugar. This is your pentose sugar attached to a nitrogenous base and phosphate groups. Now, this pentose sugar can be ribose sugar or it can be deoxyribose. Now, this nitrogenous base can be guanine, it can be adenine, any base. It can be your uridine. Okay, now these phosphate, it can be monophosphate, only one phosphate here. It can be diphosphate, two phosphates, as in this case. So here you have uridine and you have a sugar and you have two phosphate. So this is your uridine diphosphate. Okay, this is a nucleotide. Now to this uridine diphosphate, what happens is glucose is attached here. So this forms a UDP glucose, which is a nucleotide derivative. Okay, so your UDP glucose. Now it is saying that the alpha D glucose attached to uridine diphosphate is the source of all the glucosyl residues that are added to the growing glycogen molecule. So this UDP glucose is the source of glucose that will be added to your growing glycogen chain. Okay. Now, glucose by hexokinase changes into glucose 6-phosphate, which changes to glucose 1-phosphate by phosphoglucomutase. Okay, so what it's saying is, yeah, your glucose, it changes into glucose 6-phosphate. This is brought about by enzyme hexokinase. 1 ATP is used in this reaction. We've also spoken about this reaction in glycolysis as well. So, hexokinase, what it does is it transfers a phosphate to glucose to form glucose 6-phosphate. Okay. Now the next step is this glucose 6-phosphate changes into glucose 1-phosphate. This is brought about by phosphoglucomutase. This is an isomerization reaction because the phosphate which was attached to position 6 goes to position 1. Now what happens is this glucose 1 phosphate, it combines with UTP. Now you very well know what is UTP. I just explained this. This will be your UTP, okay? A nucleotide here. So your glucose 1 phosphate, it combines with a UTP to form your UDP glucose. So you get your UDP glucose. Now the catch point here is, and this is brought about by which enzyme? This. The enzyme used is UDP glucose for pyrophosphorylase. Okay, this is the enzyme used. But have you noticed something here? That your substrate here has 1 phosphate and this has 3 phosphate. So glucose 1 phosphate, it has 1 phosphate. UTP, it has 3 phosphates. So your substrate had a total of 4 phosphate, but your product UDP glucose diphosphate, it only has 2 phosphates. So your 2 phosphates are released. That means 2 phosphates were released as pyrophosphate in this reaction. Okay. Now we've seen the synthesis of UDP glucose. Now this was the first step in glycogen synthesis. Now moving on to the next step, which is catalyzed by glycogen synthase. This is the rate limiting enzyme of the reaction. Now glycogen synthase reactions. Glycogen synthase is able to make alpha 1,4 linkages. This. It cannot initiate chain synthesis using free glucose as an acceptor of a molecule of glucose from UDP glucose. Glycogen synthase needs a primer. Now, this is something very important. It says that your glycogen synthase, which makes alpha 1,4 linkages, it cannot initiate a reaction by adding those glycosyl residues that were coming from UDP to a free glucose. It needs a primer to work. Now, we will see what are those primers that are needed by glycogen synthase. A fragment of glycogen can serve as a primer, or something known as glycogenin can serve as a primer. Now, what is glycogenin? Sometimes when the cell is not completely, has not been completely depleted of its glycogen reserves, a fragment of glycogen is left, that can serve as a primer. And when that a fragment of glycogen is not present, in that case, your glycogenin is what is a glycogen primer. Now, what is glycogenin? Glycogenin is a homodimeric protein. Okay, so it is a polypeptide. Your glycogenin is a protein and it has 2 identical monomers. An oligosaccharide chain of 7 glucose units is added to each monomer. So your glycogenin is something like this. 2 same identical monomers are there to form glycogenin, and this glycogenin is going to serve as a primer for the synthesis of glycogen. Now, your glycogenin is rich in tyrosine residues. Okay, so the side chain of tyrosine has a hydroxyl group. So, side chain of hydroxyl group of tyrosine 194 is the site. In the protein is the site at which initial glucosyl residue is attached. So, it is saying that there is a primer known as glycogenin. It is a protein, and this protein is rich in tyrosine residues, and the hydroxyl of the tyrosine is the first site where glucosyl residues will be added. So glycogen primer is formed by autoglycosylation of glycogenin. Monomers of glycogenin glycosylating each other using UDP glucose until 7 residues are added. This short chain acts as glycogen primer that is elongated by glycogen synthase. Okay, so I'll explain you this a little. This is your glycogenin primer made up of 2 identical units, and it has tyrosine residues. The hydroxyl of the tyrosine is the site where your glucosyl residue will be added. But how is the primer formed? It is saying that both the identical units, they glycosylate each other until 7 glucose residues are added. 1, 2, 3, 4, 5, 6, 7. So until this structure is formed, they will keep glycosylating. So both the monomers, this is one monomer, there must be another monomer. This will glycosylate this, and this is autoglycosylation. For this glycosylation, that is addition of glucose onto the protein, no other enzyme is needed. The monomers are autoglycosylating each them. One monomer is glycosylating the other one, and the other one is glycosylating this one. So glycogenin itself is acting as an enzyme. You will study about this in signal transduction as well. The tyrosine kinase, it autophosphorylates it, and like here it is glycosylating, adding carbohydrate glucose, there it will phosphorylate, autophosphorylate itself by adding phosphate group. So now your primer is ready. This is your glycogen primer and it is ready. Now on this primer, elongation takes place by glycogen synthase. Okay. From now on, I'll be drawing the primer something like this. Okay, I'll denote the primer with a green dot just for the sake of convenience. But you know by this green dot what I mean, that this glycogenin, it is a protein made up of two homodimers, and then they autoglycosylate each other to form a 7 glucose long oligosaccharide. Okay, and that entire structure is known as a glycogenin primer. On this primer, now glycogen synthase will begin the synthesis of glycogen. Okay, now let's see what happens, how the elongation takes place. By the action of glycogen synthase, glucose is transferred from UDP glucose to glycogenin. Glycogen synthase makes alpha 1,4 linkages. The addition will take place up to the length of 11 residues. So a linear polymer has been synthesized. So it is saying something like this. For the sake of convenience, I'm just denoting glycogenin like this. Okay. Now on this primer, which is your glycogenin primer, what will act? Glycogen synthase will act. Okay. Glycogen synthase will act, and from where the glucose residues are being added? From UDP glucose, which was the first step. We've learned this step. So glucose are being added to this primer an n number of times. N by n, I mean up to like to the length of 11 to 12 residues. So you will get something like this. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay. So you get a linear structure, a linear polymer. But this linear polymer is not glycogen, because glycogen is branched. This is linear. And glycogen synthase is able to make only alpha 1,4 linkages. Okay. So these linkages here are alpha 1,4 linkages. And this is exactly what we just read. That by the action of glycogen synthase, glucose is transferred. You can see glycogen synthase is acting. Glucose is being transferred to the glycogenin primer up to the length of 11 residues, making alpha 1,4 linkages. And a linear polymer has been synthesized. Okay. Now, but as I said, that glycogen is branched, so we need to make branches. Now comes the branching. Branching enzyme creates alpha 1,6 linkages. When the chain is 11 to 12 residues long, the branching enzyme transfers a block of 6 to 8 residues from this chain to another site on the growing molecule. This is the name of the branching enzyme. What is this? Amylo alpha 1,4,1,6 transglucosidase. Okay, now again I'll write and show you what it says. This was your in green color. Okay. This was your primer. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay. Now it's saying a 6 residue block is transferred. And this is brought about by glucan transferase, alpha 1416 glucan transferase. It will transfer. See, it is saying when the chain is 11 residue long, the branching enzyme, now comes the branching enzyme. The name of the branching enzyme has been mentioned. I'm just quickly writing branching enzyme, okay? Okay, now comes the branching enzyme. What will the branching enzyme do? That it will transfer a block of 6 to 8 residues from the growing chain. This was your growing chain. A block has been transferred here to another point, okay? This creates a branch. So what will happen? It will look something like this. In red color. Okay, so these molecules have been transferred here. And what is this here now? This is the branching point, and this branching point, it has alpha 1,6 linkage, and this is made by the branching enzyme. And what were these were alpha 1,4 linkages, and they were made by glycogen synthase. Now this process will keep repeating again. Now glycogen synthase will act and make it up to the length of 11 residues. Then again branching point will act, then it will transfer it somewhere else, like this, and this process will continue, and more branching will occur, and you will get your glycogen. So that was all about glycogen synthesis. I hope it was clear. I'll quickly recapitulate the steps. What were the steps involved? Synthesis of UDP glucose. How UDP glucose was synthesized, we just saw here. This is how your UDP glucose is synthesized. The glucose, it changes into glucose 6 phosphate, then to glucose 1 phosphate. Now this glucose 1 phosphate, it combines with UTP to form a UDP glucose. Now this UDP glucose is the donor of glucose in glycogen synthesis. Now what happens is, you need a primer. What was a primer? Glycogenin was a primer. What is glycogenin? We discussed. Now on this primer, the glucose from the UDP will be added by glycogen synthase and make it 11 residue long. Then after that comes the branching point, which will transfer a 6 residue unit from the end to another point and create a branch. And the branch has alpha 1,6 linkages. Okay. Now moving on, let's begin with glycogenolysis. This is breakdown of glycogen. Okay. It takes place during early fasting. Note this. The site is liver and in the muscle. The organelle is majorly, it is your cytoplasm, but 1 to 2% of it takes place in the lysosome as well. And smooth endoplasmic reticulum. The rate limiting enzyme is glycogen phosphorylase, and the coenzyme for this is PLP, pyridoxal phosphate, which is your vitamin B6. Now the steps in glycogenolysis are action of glycogen phosphorylase, removal of the branches, and conversion of glucose 1 phosphate to glucose. So we'll discuss each of these steps one by one. Action of glycogen phosphorylase. Glycogen phosphorylase cleaves alpha 1,4 linkages sequentially from the end of the glycogen branch up to the length of 4 glucose units away from the branch point. Okay, let's see what this is. this was your primer and one, two. Now, this is, for example, your glycogen. Now, this glycogen is acted upon by glycogen phosphorylase. So, your glycogen phosphorylase, it will cleave glucose 1 phosphate residues. Okay. So, it will give out glucose 1 phosphate residues to form a product which looks something like this. Now, the catch point here is, see what it says, 4 glucose residues away. Okay, so your glycogen phosphorylase will work only 4 glucose residues away from the branch point. You see, this was the branch point here. So it is cleaving glu glucose 1 phosphate residues from the end until 4 glucose units away from the branch point. And also another thing to be noted is that it is removing glucose as glucose 1 phosphate and not as glucose. So the final product is a highly branched molecule which is known as limit dextrin. This is your limit dextrin. Because the branches have not been removed, so it is highly branched. Let's read this again. Glycogen phosphorylase is your enzyme. It cleaves only alpha 1,4 linkages. Okay, so it is not capable of removing the branches sequentially from one end of the glycogen branch up to the length of 4 glucose units away from the branch point. So you see, this was your glycogen. It was acted upon by glycogen phosphorylase and it sequentially removes glucose as glucose 1 phosphate and it works only up to the length of 4 glucose units away from the branch point. Okay, I hope this clears. Then the question arises how the branches are removed. So this is the second step, how your branches are removed. So this is done by an enzyme known as debranching enzyme. Okay, so this is brought about by an enzyme known as debranching enzyme. It is a bifunctional enzyme. Now, what is a bifunctional enzyme? Bifunctional enzymes are those enzymes which have two enzyme activities. That is, your same polypeptide chain will be having two different enzyme activities. That is a bifunctional or a tandem enzyme. So your debranching enzyme, the enzyme which removes the branches, it is a bifunctional enzyme. And what are those two enzyme activities? It is oligo. This is alpha, okay. Oligo alpha 1414 glucan transferase activity and amylo alpha 16 glucosidase activity. First, okay, let me make it more clear for you so that you understand. This is your bifunctional enzyme. It has two enzyme activities, alpha 1414 glucan transferase. I'm not writing the whole thing. And the other one is amylo alpha 16 glucosidase activity. Now what happens is you had your limit dextrin here. So this will work on this limit dextrin. How does it work? That it first you will have the glucan transferase activity. See, first action of debranching enzyme is that is alpha 1414 glucan transferase will be active. This one, it cuts an outer trisaccharide unit and relocates it to the non-reducing end of the other chain. Thus, an alpha 1,4 bond is broken and an alpha 1,4 bond is made. Okay, so what it means is This was your limit dextrin. On this limit dextrin, your debranching enzyme will work, and debranching enzyme is a bifunctional enzyme, has two enzyme activities. So the first action is by glucan transferase. So what will glucan transferase do? That it will transfer this. It will transfer a trisaccharide unit. Okay, 3 units. It will transfer this 3 unit and relocate it. So what will happen is this will look something like this. Now this will use your glycogen. These 3 units are transferred, so they move here. And now what are you left with? You're just left with one glucose here. Okay, but the branching point, you see, the branching point is still intact. So this was the first action of your debranching enzyme. Now the second action of debranching enzyme is the alpha 1,6 linkage is broken. Now is when your alpha 1,6 linkage, that is the branch point, is broken, and this is done by amylo alpha 1,6 glucosidase activity and released as free glucose. Okay, here now this is acted upon by amylose alpha 1,6 glucosidase. Oh, where it is? Yeah, glucosidase, amylo alpha 1,6 glucosidase activity, and this will release your glucose as glucose only. So you will end up with plus glucose. So the glycosyl chain is now available again for degradation by glycogen phosphorylase until 4 glycosyl units in the next branch are reached. Major product of glycogenolysis is glucose 1-phosphate and not glucose, as glycogen phosphorylase is acting mainly and alpha 1,6 glucosidase act only at branch points. Okay, this is very much understandable. See this diagram again. This was your glycogen. Glycogen phosphorylase acts on it and removes sequentially glucose as glucose 1-phosphate. Then you end up with a structure which is known as limit dextrin, and it is highly branched. Then comes your debranching enzymes. It is only cleaving alpha 1,4 linkage, which is your glycogen phosphorylase. Now you end up with limit dextrin. Now this is acted upon by bifunctional enzyme, which is the debranching enzyme. The first activity is that it transfers a trisaccharide unit and relocates it to the end, other reducing end of the glycogen. Now you left with just glucose and the branch point. So this branch point is removed by amylo alpha 1,6 glucosidase, and then you end up with glucose. Free glucose has been removed. So majorly glycogen phosphorylase is acting, so you are getting glucose 1-phosphate, and majority of it is glucose 1-phosphate, and you're getting minor amount of glucose. Now up till here, in liver and in the muscle it was same. Glycogenolysis up to this step it was same in the liver and the muscle, but from now on there's difference. We'll see what the difference is. And why do you need to convert glucose 1-phosphate to glucose? Because in the blood you need glucose, you do not need glucose 1-phosphate. Okay, so why? Because blood needs glucose and not glucose 1-phosphate. And secondly, all this happens inside the cell, and glucose transporters can only transport glucose. Okay, so you have glucose transport, and you already know I discussed this in glycolysis also the first time, that this is trapping of glucose. So when you attach phosphate to glucose, it is trapped inside the cell, and you do not have transporters for glucose 1-phosphate. You have transporters for glucose. So how do you take that glucose 1-phosphate out of the cell? So that is also one reason why you need to convert glucose 1-phosphate to glucose. And also because your body needs, your blood needs glucose and not glucose 1-phosphate. And glucose 1-phosphate as such cannot move out of the cell because you do not have transporters for it, and it is too polar to move through the lipid layer. Now what happens is in liver, glucose 1 phosphate changes to glucose 6 phosphate by phosphoglucomutase, which then changes to glucose by glucose 6 phosphatase. Okay, so it says to glucose 1 phosphate, it changes to glucose 6 phosphate. And this glucose 6 phosphate changes to your glucose. Now, glucose 1 phosphate changing to glucose 6 phosphate. So, what is this? Isomerization reaction. So, mutase enzyme is involved because from position 1, the phosphate goes to position 6. So, there is only change in the position of phosphate group within the same molecule. So, a mutase is required. Now, this glucose 6 phosphate, it changes into glucose. This is done by glucose 6 phosphatase enzyme. And your phosphatase is a hydrolase, so it breaks covalent bond by adding water, and an inorganic phosphate is released. So you see your phosphate is removed from glucose. Now this glucose can move out of the cell. The glucose transporters are bidirectional. When glucose is more in the blood, it enters the cell. When the body needs glucose, you are in starving or fasting state, then it can very well provide with glucose as well. So now this glucose can move out of the cell into the blood and maintain the blood glucose level. Now, do you understand the first point in the beginning in the introduction when I said that the liver glycogen aims at maintaining the blood glucose level? Okay, so this is how your liver glycogen helps in maintaining blood glucose levels. Now, see what happens in the muscle. In the muscle, glucose 6 phosphatase is absent. Thus, glucose 6-phosphate enters anaerobic glycolysis, providing energy needed for muscle contraction. So, a liver is a very selfless organ, but muscle is a selfish organ. Okay, it uses this for its own need. It uses glucose 6-phosphate to enter anaerobic glycolysis to generate ATP energy for the sake of muscle contraction. Because in muscle, the enzyme glucose 6-phosphatase here, this enzyme glucose 6 phosphatase responsible for converting glucose 6 phosphate into glucose is absent. This is in the muscle. So in the muscle, liver glycogen in the muscle to glycogen, that is muscle glycogen, is used to provide ATP for muscle contraction. But your liver glycogen, that is glycogen in the liver, is used for maintaining blood glucose. So in this case, you have glucose 6 phosphate entering anaerobic glycolysis. And in anaerobic glycolysis, for reference, you can see the previous video, you have production of 3 ATP. But in this case, since the first step is absent, hexokinase step is absent, so you will be using 1 ATP less and generating 1 ATP more. So the net yield is 3 ATPs as the hexokinase step is not there because it is entering directly at the level of glucose 6 phosphate. Now let's discuss few points about the enzyme glucose 6 phosphatase. It is present in liver, but it is absent in muscle. It is found in endoplasmic reticulum. It is common to both gluconeogenesis and glycogenolysis. When we study gluconeogenesis, you will see, because in the last step, when you're forming glucose in gluconeogenesis, you need glucose 6 phosphatase to convert the glucose 6 phosphate into glucose. This glucose 6 phosphate in the cytoplasm is transferred into the endoplasmic reticulum by a transporter known as glucose 6 phosphate translocase. Okay, this is the reason because your glucose 6 phosphatase is present in endoplasmic reticulum and not in the cytoplasm, because we discussed that all the enzymes for glycogen metabolism are present in the cytoplasm. but your glucose 6 phosphatase is present in the endoplasmic reticulum. Therefore, your glucose 6 phosphate is not immediately changed into glucose, because your glucose 6 phosphate is first transferred to endoplasmic reticulum, then it is acted upon by glucose 6 phosphatase, and then it is changed into glucose. And then this glucose from the endoplasmic reticulum moves out into the cytoplasm and then out of cell. Then it is dephosphorylated to glucose by enzyme glucose 6 phosphatase. This glucose then goes out of the endoplasmic reticulum via glucose transporter GLUT7. So it is trying to say something like this. Like this is your endoplasmic reticulum, and this is a transporter translocase. So your glucose 6 phosphate will enter into the endoplasmic reticulum via this transporter. And here in the endoplasmic reticulum, you have glucose 6 phosphatase enzyme. So this glucose 6 phosphate will change into glucose by the action of this enzyme. And then this glucose via GLUT7 will move out from the endoplasmic reticulum. Okay, that was all for today. Thanks for watching. I hope you enjoyed the lecture. If you have any queries, I'm right here. You can ask. Um, let's take up some questions. Which of the following tissue glycogen is incapable of contributing directly to blood glucose? In which of the following tissue glycogen is incapable of contributing directly to blood glucose? Okay, we just discussed this. Muscle. Muscle uses its glycogen for the purpose of generating ATP. So the answer will be, which of the is incapable of contributing? It will be muscle. Okay, the next question. So obviously it will be the enzyme which is common to both the cycles. Why glucose 6 phosphate in the cytoplasm of hepatocyte is not acted upon by glucose 6 phosphatase as soon as it is formed? And now we have the last question of the quiz. Glycogen phosphorylase coenzyme is. Okay, so that was a good response. Almost everyone answered all the questions correctly. So if you have any queries, you can send.